Coming to the discussion, the study shows that the cranial sutures harbor skeletal stem cells whose equilibrium is critical for maintaining the suture patency. A marked decrease of these cells is observed in both physiologically fusing and pathologically syndromic or non-syndromic craniosynostosis. The transcriptomic analysis revealed distinct molecular signatures in progenitor cells from fusing versus the patent sutures, suggesting transcriptional priming towards osteogenic or chondrogenic fates. The WNT signaling was identified as the key regulator. Activation expanded progenitor cells and preserved patency, while the inhibition depleted them and induced fusion. The functional in vivo test showed that combining the stem cells along with the WNT3A protein could rescue craniosynostosis and prevent re-synostosis after surgical suturectomy. Also, the findings highlight the therapeutic potential of stem progenitor cells based and WNT-driven approaches in managing craniosynostosis. So, the strength of this article, firstly, it demonstrated skeletal stem progenitor cells dynamics with cranial suture patency and craniosynostosis. It uses complementary approach like FSC's bulk single cell and bulk cell signaling, histology, in vivo transplantation, and genetic mouse models. And they also provide a functional full proof of concept that WNT activation and cell transplantation can restore and maintain suture patency. The study offers translational insight that may guide regenerative or molecular therapies for craniosynostosis. The limitations: the sample size was not statistically predetermined and raising a potential bias in the effect estimation. The findings are restricted to mouse models and limiting direct clinical translation to humans. The dependence on exogenous WNT3A suggests complexity in clinical application compared to the other stem cell populations. And the functional heterogeneity of subpopulation identified in single cell RNA sequencing was not fully resolved. So this study followed the arrived guidelines where the sample size was partially followed, as I said before, and the inclusion-exclusion criteria was not explicitly mentioned, whereas the randomization and binding was also not described. Lastly, the protocol registration was not mentioned. The rest, the guidelines were followed. So that's the article. Thank you.